So this is our isolation and comparative genomic characterization of Streptomyces, Bacteriophages, Katalie, and South40. So bacteriophages are specific types of viruses that infect and replicate only in bacterial cells and are made up of nucleic acids protected by a protein coat. Bacteriophages, also known as simply phages, significantly outnumber prokaryotic and eukaryotic species and express genetic diversity that has only just begun to be analyzed, being recognized as the most abundant biological agent on Earth. Phage genome sequencing, which involves determining the precise order of nucleotides to determine which genes encode which proteins, is becoming increasingly more important in microbial research and is providing promising preliminary insight into the use of phage for therapeutic treatments against diseases caused by their bacterial hosts. Cphages is an international project designed to analyze individual phages within the bacteriophage population and gain insight into the evolutionary processes that resulted in this enormous and diverse microbial community. At the forefront of Cphages research are phage cocktails, which are mixtures of different phages that are designed to kill disease-causing bacteria. Determining the mixture of phages that will most effectively kill the host is done by analyzing the similarities and differences between phage genomes and the proteins they encode to see how different bacterial strains will respond to the presence of different phages. As a result, the overall purpose of our research is to see how our phages, Katalie and South40, fit within the context of phages already sequenced to advance the use of phages in medical applications. So just some big picture details of Katalie's genome. So before beginning any genome annotation, FAMReader showed that Katalie had 78 potential gene candidates, and we were able to narrow these down to 72 true genes that we were going to be working with. Phage Katalie is in cluster BD and subcluster BD1 with a 3' sticky overhang end structure. Katalie has a total genome size of 51,271 base pairs with a 65.6% GC content. So just an overview of the methods that we went through in the fall of 2020 and spring of 2021 to get where we currently are with genome annotation. So we first had to isolate and purify phage Katalie to ensure that we were working with the clonal population of phage. So we first did so by collecting a soil sample from Forest Park in St. Louis, Missouri, and we enriched the sample to amplify phage growth before we ever plated it. We then purified this enriched sample by plating it on agar plates and letting the plates incubate, and we also diluted the phage samples with each of our 5 rounds of purification. By performing plaque assays after each incubation period, we were able to identify that we did have true plaques, and we then created a high titer lysate by flooding the webbed plate that we created with phage buffer, and we then collected this liquid sample so that we had a high concentration of to work with in our genome analysis. Throughout isolation and purification, we documented the appearance and presence of plaques with each round of dilutions, and we also obtained electron microscopy images to see Katalie's protein structure, and we wanted to use this structure to compare Katalie to other phages in cluster BD to see how similar or different they were. And finally, in the spring of 2020, we began our genome analysis by first performing positional annotation to identify the start nucleotides of each respective gene. And then with these starts confirmed, we moved on to functional annotation, which is where we currently are in our genome analysis. So in order to identify gene function, we first determined whether we could identify a putative function, no known function, or whether we could not assign any putative function. And at this point, we are working with HH-pred partial alignments, BLAST-P, and Synteny to further analyze any genes that we don't yet have confirmed, and we are moving into in-depth analyses of especially difficult genes.
So phage Katalie was collected on September 26, 2020 in Forest Park. The sample was collected about 6 centimeters underground and was found underneath a tree by Art Hill. And the soil itself was very dry and slightly sandy. This sample was later enriched and it underwent 5 rounds of purification. This purification process included both modified and formal plaque assay procedures with serial dilutions. In the first 2 rounds, we were able to verify the presence of phage in our sample, whereas in later rounds, we ensured that we only had 1 bacteriophage isolated. Following this, a webbed plate was made, which we can see on the next slide. So as I mentioned, we created a webbed plate, which allowed us to collect a high titer lysate. So the images on the left are showing the top and bottom view of this webbed plate. And we came to the conclusion that our 10 to the -6 dilution, which is shown on the right, was ideal for plaque presence. Using this 10 to the -6 dilution, we calculated a final titer lysate of 4.2 times 10 to the 8th plaque forming units per milliliter, which represents the concentration of phage presence in the high titer lysate. And as you can see on the right, the plaques did vary a little bit in size, but they were very clear and very distinct from each other.
Next, from our high titer lysate, we used electron microscopy to determine the structural morphology of bacteriophage Katalie. And we can see in these images that bacteriophage Katalie has long, flexible tails and capsids that are almost circular. In fact, some of these look to be slightly icosahedral, but almost all of them are most definitely rounded. From these results, we were able to come to the structural morphology that Bacteriophage Katalie is a Cephuviridae, and this is very similar to the structural morphology of other phages present in its cluster and subcluster. Additionally, from our high titer lysate, we were able to isolate the DNA and then sent that purified sample off to be sequenced at the McDonald Genome Institute at the Washington University School of Medicine, and then we assembled this sequence result and were able to evaluate our genome. We then began to evaluate the putative translation starts to determine the position of each gene. Starting with 78 genes predicted by software, we determined that 6 of those weren't able to code for any protein. Phage genomes are incredibly dense, and so genes are going to, with extreme overlap, are pretty suspicious. And so we then analyzed those genes using BLAST, Basic Local Alignment Search Tool, and host-trained GenMark coding potential to determine if these were actual genes or if they weren't. And so we concluded that we only had 72 genes present in the Katalie genome. Next, we took a look at the possible translation starts. And we used tools such as Glimmer and GenMark, which are algorithms that predict the starts of translation. We looked at the gap between these because we're trying to minimize that, as well as the Shine-Dalgarno score, which will determine the efficiency of the ribosome binding to that area, as well as using the host-trained GenMark coding potential to see where the protein could be coded for, as well as BLAST to see high-quality alignments with real proteins, and Starterator, an evolutionary tool to see conservation of starts within the subcluster. Each gene underwent at least 2 rounds of analysis, with some more obscure genes needing up to 5. In addition, genes 18 and 39, which code for tail assembly chaperones and DNA primases respectively, are known to have program translational frameshifts, meaning that there's going to be a significant overlap with the genes before them, and these are going to be analyzed in some of the final steps of our analysis, which we'll do in future studies.
And so here's the table displaying our current progress with functional annotation. So by analyzing the strength of HHPRED, phages DB BLAST, and TMHMM evidence, we assign putative functions to our genes. So out of our total 72 genes, we have 55 genes with confirmed functions, which are shown in green, along with their specific roles. Highlighted in yellow are 14 genes without enough evidence to call a putative function, but have some evidence suggesting that there is a known function. In blue, we have 3 putative functions that are yet to be confirmed by solid evidence. And so moving forward with all the evidence that we have gathered from isolating and analyzing our phage, it adds to the growing scientific knowledge on bacteriophages. Specifically, in regards to evolution, our gene functions may indicate the importance of some conserved functions in specific regions of genes particularly significant for performing those functions. We also isolated another phage called South40, which differs by a single nucleotide substitution. By comparing their genomes, we may discover more about the significance of the substitution in the conservation of nucleotide sequences. Additionally, with the knowledge we currently know about the lytic life cycle, shown to the right, bacteriophages are investigated for their potential role in combating antibiotic resistance in bacterial infections, a growing problem in the modern era. Phages undergo a process called the lytic cycle, where they infect a bacterial host to replicate and build enough to cause cell lysis, which makes them a key factor when studying the immune response. Currently, bacteriophage treatments are used as a last resort in patients with cystic fibrosis, and case studies have been performed successfully.
Continuing this discussion and the comparison with bacteriophage South40, comparing specifically the methods, we can see that South40 used pretty much the exact same methods as Katalie for its isolation and purification. The only difference was that South40 used direct isolation instead of enriched isolation, which essentially means we isolated the bacteriophage directly from the soil instead of growing the phage in a suspended bacterial culture. In addition, looking at the morphology of South40, we can see that it forms medium-sized, slightly turbid plaques, as well as having a very flexible tail and an icosahedral head. Now, this actually differs a little bit with bacteriophage Katalie, which provides some interesting conflict. because Katalie had clear small plaques and a circular head. But this is likely due to the density of the plaques in the plating of South40, as well as the conditions of the electron microscopy. Where the real difference and where the real importance comes is looking at this genomic analysis and how the difference between bacteriophage South40 and Katalie is present at a difference, the difference at position 20,528, which is present in a minor tail protein in Katalie. Using comparison with BLAST, we were able to find that a guanine present in Katalie was switched to an adenine in South40. And this resulted in the conservative replacement of an aspartic acid residue with an asparagine in this protein. Both of these are polar and are capable of engaging in hydrogen bonding, so there's not likely to be a major difference between the structures and therefore functions of this protein, but using some future analysis and the FIRE2 algorithm, we're going to try and predict the structural changes that could occur as a result of this and determine the true extent to which this mutation has changed the protein. But overall, South40 and Katalie are almost identical, which helps us to provide additional evidence for the functions of each of the proteins of South40, which we will continue to analyze in our future studies. We would also like to thank everyone who helped us with this project, and especially to Dr. Hafer and Dr. Schaefer, who are our mentors throughout this entire past year. In addition, we'd like to thank the TAs for our course, Lavinia, Ala, and Bijan, as well as the Department of Biology here at Washington University, the Hatful Lab from the University of Pittsburgh, the Howard Hughes Medical Institute, and all of their support for the C-Phages program. We thank all of these people for their both assistance with our study, as well as with their support for bacteriophage research, and hope you all have a great rest of your day. Thank you.